Hello, everyone, and welcome to today's webinar, Interrogating 3D Spheroid versus 2D monolayer cell models in drug discovery using cell health microplate assays. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit www.thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want and any time you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Dr. Adyary Fallarero, Senior Scientist at Thermo Fisher Scientific, and Dr. Leticia Montoya, Scientist, Cell Biology at Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations.
Well, thank you so much, Michelle, for the introduction. My name is Adyary Fallarero, and along with my colleague Leticia Montoya, we are going to be presenting today a number of results that will reveal to you what are the main critical points that you have to be taking into account when you are transitioning between 2D and 3D cellular models, and especially when you are utilizing cell health microplate assays. So we are going to focus today in trying to reveal to you what are the critical points that you should be taking into account. The presentation will be divided into two parts. So during the first part of the presentation, I will be discussing with you guys several key issues that has to be taken into account when performing this kind of side-by-side comparison, and simply when moving from one cellular system to the other one between 2D and 3D systems. And I'm going to give you an overview of all the available products and instruments that Thermo Fisher Scientific has at your disposal to facilitate this transitioning. And then we are going to focus on some of these theoretical aspects that are really important to be able to interrogate cellular health in 3D. So what are the critical issues? What would be important to measure? How such measurements should be performed, especially when utilizing microplate readers. And then my colleague Leticia will go over through 3 different cases of cell health assays that have been developed originally for 2D cellular systems by Thermo Fisher Scientific, but we are hoping now that we can demonstrate to you guys how those systems can be utilized also on 3D. We are going to show you results that will illustrate both endpoint and kinetic assays. And then, last but not least, we are going to give you some ideas and really showcase how this quantitative approach utilizing microplate readers can be really very well correlated with the results that can be obtained with high content imaging. Hopefully, after this hour of discussing with you guys and giving you our thoughts, then you will be able to learn some important messages and be able to really take those messages and facilitate your work in the future. So I'd like to start today by going over through something that is probably quite familiar for you all, and it relates to the pipeline of drug discovery. We all know that the pipeline of drug discovery is characterized by a quite strong emphasis on throughput at the very early stages of the drug discovery process. At the very early stages of the drug discovery process, scientists are interested in really generating a large amount of promising leads that can be then further developed into promising drug candidates. So for these high throughput analysis at the very early stages, we have traditionally utilized in vitro assays, and that has been routinely performed in 2D. And the reason why they have been performed in 2D is because such models are extremely user-friendly from the perspective of the throughput. So they are very simple models, and they are very easily adaptable to high-throughput screening conditions. However, the main detriment or the main drawback of such model is they are really restricted simplicity and the fact that they often lack on predictive capacity. So in an attempt to really improve this quality of the in vitro screens that are performed at the early stage of the drug discovery process, so there has been a progressive shift towards the utilization of 3D cellular systems. And this is perhaps a good moment for you guys, for us to clarify that often in the literature we talk about 2D and 3D, but we are in both cases talking about cells, and they, of course, they are all in 3D. They exist as in 3D. But when we talk about 2D, we talk about adherent or suspended cells, and they are dramatically different from the cells that are growing in a more geometrically structured shape that we will shortly discuss afterwards. For the purpose of this presentation, every time that we talk about 2D, we refer to monolayers, cellular monolayers, and every time that we talk about 3D, we are focusing on spheroids. So really the fact that with spheroids we can still maintain a very high throughput while gaining a valuable complexity, it's really what has prompted an explosion of 3D studies. You can see at the moment quite close to 1,000 peer-reviewed publications published every year on the topic of 3D. And well, this is the reason why we are discussing today about 3D. We are trying to aid this transition. We are trying to help you guys using new tools that can be utilized for this transition. So the question that I want to tackle now at the next is, why do we believe that these 3D systems are actually better models? Why do we believe that this added complexity actually increases the predictability value of those systems? And really, the main feature that it's very relevant to indicate for spheroids in general for 3D models is that they really are better pharmacological models, and there's been an increasing amount of evidence showing that you get much better pharmacological predictability with these 3D cells. The main reason behind that is simply that when 3D cells are formed, when spheroids are formed, you end up with an ecosystem of cells that are interacting with each other in such a way that really resembles much better what happens in vivo, especially in the case of solid tumors. So 3D cells, and especially spheroids, have become much better and much more advantageous models for pharmacological prediction of drugs in solid tumors. They are better models of in vivo hypoxia. They are better models also of this in vivo heterogeneity that is typical of tumors. And you can really study the effect of drugs in a much more valuable way, a much more meaningful way that we could in the traditional 2D monolayers that have been utilized in drug discovery. And so as we move from 2D to 3D, we often encounter scientists that are developing new 3D systems, but when developing these new 3D systems, they are often in this situation that they need to utilize or they need to interrogate cellular health functions, and they are unable to find the tools that can be utilized simultaneously in 2D and 3D models. The truth is, as you can see in this comparison table, that the majority of the commercial and even in-house, let's say, workflows that exist currently, they are built around 2D cultures, and scientists are faced with the situation that they just don't have enough tools, especially commercial tools that can be utilized readily from 2D to 3D. So this is a situation that we are trying to tackle and that we are trying to address here at Thermo Fisher Scientific. So our R&D scientists are trying really to validate better tools. We are trying to really address the lack of these validated assays. So this is precisely what we are going to try to illustrate to you guys today. We are going to start with the current portfolio of products that we have to support 3D cellular models. I must mention also at this stage that we are not going to discuss today anything related to how 3D cell cultures are grown and how they are formed. we are only focusing on how to interrogate 3D cellular functions once these 3D cells or 3D cell systems have been developed. But it's good for you guys to be aware that we offer a very broad portfolio of products that includes cells, that also includes matrices and cell culture vessels. And especially in the case of scaffold-free 3D systems, Thermo Fisher has developed the Sphera portfolio of products that covers a number of different cell culture dishes and cell culture vessels that have ultra-low attachment surface. And this is precisely these Sphera microplates are the ones that we are going to be showing today in our results. We also have a number of media and supplements that can be utilized for growing cells in 3D. And last but not least, we have a number of instruments and assays and reagents that can be utilized to study cellular functions in 3D. And those are the ones that we are going to be focusing on the presentation today. But as I said, we are here to help you. So if you have any questions about any of these products that you have seen here in this presentation, please reach out to us. And if we cannot answer the question right away, then we will be happy to put you in contact with one of our specialists. So when we talk about approaches that have been utilized to study 3D, it's very, very often the case that we see researchers utilizing high content imaging and fluorescent imaging. Those have been traditionally used for exploring 3D morphology, for studying 3D phenotypic changes. But now, more and more, we also see scientists interested in exploring cellular function and quantifying cellular function using microplate readers. This is actually a logical approach. We feel that for scientists, microplate readers are nearly in every lab, in every life science lab. So it's a very very useful shift to be able to continue using imaging, but also being able to progressively adapt more and more cellular assays that are so widely available for 2D models into the 3D models space. So today we are going to focus on quantification of cellular function using microplate readers. And the microplate reader that is our hero today, it's the Thermo Scientific BodyScan Lux multi-mode microplate reader, which was launched in 2015, and it is really a multi-technology instrument that can be used for measuring optical signals in basically any kind of microplate format from 6 to 1536 well plates. We are shortly going to be going over these detection technologies, but I want to point out that BodyScan Lux is a microplate reader that allows you to perform assays in endpoint and kinetic mode, which we will be covering today. But we can also, or you can also perform experiments in spectral, multi-point, and kinetic spectral mode. These are typically available modes for all of the microplate readers that are available in the market. So even though I will be referring often to BodyScan Lux multi-mode reader today, most of the theoretical assumptions that we are going to be discussing and most of these theoretical critical points that you have to be aware of, they would also be applicable to other multi-mode microplate readers. And last but not least, one important feature that I want to bring to your attention when it comes about BodyScan Lux is that BodyScan Lux is operated with a free software. It's called Scanit software, and within Scanit software, every user has access to an extensive protocol library, the Scanit Cloud Protocol Library, which currently contains over 130 sessions, and some of the assays that we will be discussing today are included in this protocol library. Every single protocol library, every single protocol assay in the library has real data with real results, with real data analysis, and it's not really curated data. So our scientists make the same mistakes that users do, that scientists do. So we have wanted to keep it as authentic and genuine as we possibly can. So you will find there real representative data, and you will be able to really analyze that data and explore that data to learn about the usage of our instruments and our software. So so now I said that we will now talk a little bit about the technologies that can be read with microplate readers. This is something very broadly presented, even though I'm referring to BodyScan Lux. So the majority of the multi-mode readers that are available for scientists, they are able to measure optical signals that are mostly absorbance, fluorescence, and luminescence. Bioscan logs can also measure a special type of fluorescence that is called time-resolved fluorescence, or TRF. And we can also measure another special technology that is called alpha screen. Today, we are going to be focusing on the two most widely used technologies, or at least two of the most widely used technologies, and that is absorbance and fluorescence measurements. And when we talk about these two different detection technologies, it is very, very common the case that scientists really get worried about the possible differences in the detection between 2D and 3D cellular models, and they often get intrigued about the fact that they don't know how to select which is the best possible measurement mode that they could utilize with their samples. So we are going to try to briefly touch upon the critical things that have to be taken into account when performing either absorbance or fluorescence intensity measurements. I'll start with a very simple comparison between absorbance, or that is also called photometry, and fluorescence measurements. And bear with me, I know that many of you are familiar with these technologies. I just want to give you a very brief overview so that you can understand why the way that the technology works really influences the way that our instruments are designed, and in general, most microplate readers are designed. So when we talk about photometric measurements or absorbance measurements, we are interested in measuring absorption of light. So to be able to measure absorption of light, samples need to be illuminated at a very specific wavelength where they have the maximum of absorbance, and the instrument simply record or measure the light intensity that comes out of the sample. So for the purpose of performing absorbance measurement, we have a wavelength separation step that happens before the sample is illuminated, and then we are recording all the light that goes through the sample. So in most microplate readers that are able to measure for photometry, you always have a detector and a lamp that are located in opposite sides of the plate. In the case of Bioscan logs, we are illuminating the samples from the top and then measuring from the bottom of the plate. But the sample has to be illuminated all the way through to be able to measure absorbance, or to be able to record photometric measurements. This is a slightly different situation in fluorescence. In fluorescence, compounds are also illuminated, or samples are also illuminated at a very specific wavelength, the same way that we do in absorbance measurements. But in the case of fluorescence measurements, as you are aware, the molecules that are having fluorescent properties, they go into an excited state. And after that excited state, there is a return to the ground state that is coupled with the emission of light. And that emission of light happens at a very specific wavelength. So in the case of the recording of fluorescence or the detection of fluorescence with microplate readers, there is always a wavelength separation that happens before the light hits the sample, before the sample is illuminated, and then after the sample it's illuminated, so that you are able to record the specific emitted signal that takes place at that specific excitation light. Most of the microplate readers that are out there can do that in two different ways. They can record the fluorescence that is generated from the top of the plate, and they can also record the fluorescence that is generated from the bottom of the plate. And this makes kind of a big distinction on the way that we can also measure 2D versus 3D. So what is critical to consider in the case of absorbance when comparing 2D versus 3D? So I already mentioned that absorbance measurements are always performed through the well. And when microplate readers detect the light, the intensity of the light that comes out of the sample, they really cannot make a distinction on the nature of the absorption process that took place on the sample. And what we know is that really the decrease on the light intensity that we measured with microplate readers can be caused mostly by the light absorbance that is on the sample, but it also can happen due to the light scattering of the sample. And that is that samples can really deviate the angle into which the light is actually illuminating the sample. So as said, we cannot discriminate, microplate spectrophotometers cannot discriminate between these two processes, and they are really different processes. As you can see in this scheme that it's here located in the right part of the slide, during the scattering process, particles that are on the sample are really scattering light. They are deviating the path of the light. And because of this nature of this process, because of the fact that they are deviating the path of the light, then this scattering process is strongly dependent on the distance that exists between the sample and the detector. So if we go back to this scheme to the left, where you see the differences between 2D and 3D, it can now become a little bit more obvious for you guys that if we are comparing 2D versus 3D, you are going to have a different contribution of the scattering process in 2D versus 3D, because the distance that exists between the sample and the detector will be different in the case of 2D versus 3D. So an important consideration that you have to make when performing absorbance measurements in 2D versus 3D is that you have to include controls without the dyes that can be utilized to exclude this scattering process. And really, if you see really big differences taking place in 2D versus 3D, scattering might be one of the reasons as to why this will play out. We know with Bioscan lux that the distances still between detector and sample are pretty small, and very small scattering angles will not have an impact. They will still be detected by the detector. So we know that in our instrument, in Bioscan lux, this is having still a minimal impact. But if you are using all kinds of microplate readers, this is definitely one aspect that has to be taken into account when performing absorbance measurements readouts. So then in the case of fluorescent intensity measurements, as we discussed some minutes ago, they can be performed typically in two different ways. They can be performed from the top, or they can be performed from the bottom. In the case of the top readouts, these top fluorescent measurements are ideally suited to measure cell-free systems, otherwise called as biochemical assays. You can see here in this first scheme that the optical path of Bioscan lux, which is kind of shown here with these lines and dotted lines, it always has this double conical shape. And in the case of Bioscan lux, the focal point has been designed in such a way that it hits the sample right in the middle of the solution. So when you're measuring samples that are that have fluorescence homogeneously distributed on the well, then this is an ideal optical design that will allow you to excite and measure the most of the emitted signal from the middle of the well. The situation is slightly different when we talk about bottom reading wells, so bottom reading measurements. Bottom reading measurements are generally ideal for 2D cells, especially if the fluorescence is generated inside of the cells. And that is because in the case of bottom reading, and then again, I'm referring to Bioscan lux, but it's a situation that is very similar between different manufacturers. So then in that case, the optical beam has a focal point that is exactly on the bottom of the well. So exactly in the place where 2D cells are adhering and where the most of the fluorescence will be located in the case of cell-based assays. So when comparing top reading and bottom reading, it is very generally assumed that top reading is ideal for cell-free assays or biochemical assays, and then bottom reading is ideal for 2D cells or monolayers, cellular monolayers. But then what happens with 3D cells is that with 3D cells, we fall into this kind of intermediate situation. We have still cells, and the fluorescence is often restricted to the vicinity of the cells where the fluorescent probe is actually in. But we also have cells that are in solution, that are, in the case of spheroids, that they are sort of floating in the middle of the well. So what to use in that case? It's clearly that bottom reading might not be ideal in that situation, but would top reading then be sufficient to measure the fluorescence associated to this spheroid, to this geometrical structure? Well, the answer to that question, what we have found out in our studies, is that top fluorescence can be sufficient for 3D cells, but it clearly depends on the assay, and this probably needs to be optimized for every single assay. So if your microplate reader has top and bottom reading, so a very recommended thing to do is that you would compare the performance between top and bottom reading. We also have found out that when using multimodal microplate readers, it is often recommended that one would utilize selectable excitation bandwidth. Typically, microplate readers have the possibility to switch between different excitation bandwidths, and by switching between different excitation bandwidths, you would actually would increase the beam diameter and would therefore increase the volume that is illuminated and excited on the sample. So here you see what actually happens. This is a schematic representation, but you get the point that when you are switching between 5 nanometers to 12 nanometers, in the case of Bioscan Lux, you can dramatically increase. In this case, it actually increases from 0.7 millimeters to 2 millimeters. So it's really dramatic. It dramatically increases the excited cellular volume, and therefore it increases the likelihood that you will be able to excite the cells that are located within the spheroid. So a second recommendation for you guys is that if you are able to utilize top fluorescence, then it would be recommended to use higher excitation bandwidth so that you can excite a larger amount of the cellular volume. At this point, I want to also make a clarification, because this is also a question that is often asked to us, that it is true that by increasing the excitation bandwidth using top optics, you will also end up increasing the background fluorescence. But hopefully this is something that can be taken care of, and often the increase in the background signal will not be so high that it would affect the measurement. So you will still be gaining higher signal by increasing the excitation bandwidth. But we come also across these kind of situations when scientists are comparing 2D versus 3D, and it's often the case that they are bound to use bottom fluorescence because it's simply optimal for the cell health assay that they have chosen. And if that is the case, then you have to be aware that there are differences on the way that bottom fluorescence is measured in 2D versus 3D. So when using 2D systems, 2D cellular systems, it is typically the case that cells are grown in flat bottom plates. And when bottom fluorescence is selected in multimode readers, and in particular in Bioscan Lux, so scientists tend to select these measurements using multi-points. So the measurements are performed in different locations on the bottom of the well, and you can see, as you can visualize in this slide, you can get very nice heat maps of the cellular distribution on the bottom of the well. However, if you choose to utilize this kind of multi-point measurements for bottom fluorescent readouts in 2D cells, you have to be aware that such type of measurements are not possible to perform with 3D cells, simply because if you are using a scaffold-free system as the one that we are utilizing, the Spheroid microplates, they are shaped, they have U-shaped U-bottom, and in those cases, it's not really possible to have an even illumination of the well bottom. So only the center point location can be measured on those cases. That can result in very different signals. So it would be recommendable in that case to compare only the bottom points in both 2D versus 3D. Here we have an example of what kind of impact this can actually have. This comes from our own results, and you can see here the measurement performed in 2D versus 3D with a dye that binds to the living cells on the bottom of the well. And what the impact that we can see in this case is that the signal that is recorded in 3D is higher than the signal that is recorded in 2D. And it's not really because there would be a higher amount of cells that are living on the 3D system. It's simply because the background that is measured when using multi-points across the entire bottom of the well is higher than the one that is measured only on the middle of the well in the case of the spheroids. So please be aware that when you are comparing 2D versus 3D, it is really important to select comparable patterns in the case of bottom reading, so that if you are bound to use bottom reading, then be sure that you are comparing same kind of areas, same kind of excited volumes in 2D versus 3D. Otherwise, the results are going to be impacted by this. So now we've gone through all of the most important theoretical aspects that needs to be taken into account when it comes to microplate reading, and I will continue by introducing the cell health assays that we are going to be discussing today. So you are also very familiar with this sort of a scheme that is presented here. as cells are moving from a viable to a non-viable state, they go through a number of different morphological and functional changes. And we know that in pre-lethal cells, for example, cells have an altered metabolism. They also have dysregulated cellular proliferation, very low ATP values. And all of these functional changes can be actually followed with a number of different cell health assays. When they continue to die and move to a non-viable state, then as you can see also on the scheme, then other cellular events kicked in. For example, then we have membrane permeability changes, and we have apoptosis proteins being released, and then apoptosis events being triggered. And also for those changes, there has been a number of cell health assays that has been developed. In our presentation today, we are going to focus on 3 cellular assays that are targeting metabolic studies, metabolic changes, cellular metabolic changes, and DNA content. You can see here that we are going to be talking about CyQUANT XTT, PrestoBlue HS, which are both cellular redox dye. And then we are going to be talking about the very specific dye that is used for quantification of DNA content, CyQUANT Direct. And then before I go, I want to also introduce you guys to the cellular model that we're going to be discussing today. So the cellular model that we're going to be talking about is the A549 lung spheroid model. It's a high throughput, I would say low complexity model. We are using it because it's really an excellent model. It has been extensively used for non-small cell lung cancer model, especially for the adenocarcinoma type. And it's really easy to set up when starting to perform 3D studies. We have, as I already mentioned, grown these cells in ultra-low attachment plates, Spheroplates, and we have followed the changes that take place over a number of hours. The majority of the assays that we are presenting today, they have been performed over 20 hours of spheroid growth, and those spheroids have been treated throughout the presentation with a model cytotoxic compound. It's a compound known as gambogic acid. It's not a drug, but it's a highly cytotoxic compound. And the reason why we have chosen to utilize this compound is simply because it is a very, very pleiotropic compound. So it has very many pleiotropic effects. So it affects so many of different cellular functions that it's really an excellent chemical probe and an excellent chemical tool to be able to study the impact and to be able to interrogate the different cellular functions in 3D. I want to show you now what it looks like when we have performed this kinetic imaging of this A549 spheroid. So we have visualized them using different kinds of instruments. We have utilized the EVOSM7000 Imaging System, and we have also utilized our Thermo Scientific imaging platform, the Cell Insight CX7 imaging platform. And you are going to be soon watching a video that shows how cells over time structure and become over and over more compact as time progresses. So it has been described that this is how actually spheroids get assembled. But I think it's very revealing when you actually have the opportunity to follow over time these changes that take place. And we are going to show you quickly now this video. I just want you to bear in mind that as these cells become more and more compact, they also become more resilient. They also become more chemotolerant to different kinds of treatment. And this is something that we can see from a quantitative point of view. Leticia is going to be talking over those points now, but you can also see it very nicely how they become so compact over time with this imaging system. And now you're going to see basically the results that you can, the images that you can obtain over a period of 48 hours with the Cell Insight CX7 imaging platform. And now from this point forward, I leave the floor to my colleague, Leticia Montoya, so that she will continue to walk you through these different examples of cell health assays that we presented earlier today.
Thank you, Adyary, for that introduction. Now that we have talked about the importance of 3D models and the technology used to study 3D models, we will now talk about how several existing reagents that are established for 2D models can be used for studying 3D models. Now, CyQUANT XTT is an established 2D model photometric cell viability assay that is solution-based. There are two components to this assay: XTT, which is a redox dye, and PMS, which is an electron acceptor that enhances the reduction of XTT. So this assay is an add and read assay, which means you can mix the two components together and then add the mixture to your treated or untreated cells and incubate for 4 hours at 37 degrees. After the incubation, you measure the absorbance at 450 and 660 nanometers. Now, the greater your absorbance value and your absorbance response over time, the more viable cells you have. As I already mentioned before, because this is a photometric measurement on the VarioScan Lux microplate reader, the detection method is through the well, meaning that both your 2D and 3D model systems can be analyzed the same way on the VarioScan Lux microplate reader with the CyQUANT XTT reagent. Here we have a drug dose response comparison of monolayer or 2D model systems and spheroids or 3D model systems. With this experiment, monolayer cells or spheroids were plated for 19 hours to allow the cells to adhere to the bottom of the well and to allow for a tight spheroid formation. Both of these models were then treated with gambogic acid, and cell viability was assessed with CyQUANT XTT at different hours of drug incubation time. So we looked at either 2 hours, 19 hours, or 48 hours of drug treatment. As shown within these graphs, as the incubation time increases with gambogic acid, the drug becomes more potent. Additionally, when comparing spheroids and monolayers of cells, gambogic acid is less potent to spheroids than to monolayers of cells. So within this experiment, CyQUANT XTT was incubated on the cells for 4 hours to establish a strong absorbance readout. And we can see the graphs here. On the left, you have monolayer, and on the right, you have spheroids. So here's a summary of the IC50 values obtained from the CyQUANT XTT analysis with the drug treatment of monolayer or spheroid cellular models. As I mentioned earlier, higher concentrations of gambogic acid are required to kill the 19-hour spheroids when compared to monolayer cells or to the cellular structures or cultures. So this is because of the complexity of the 3D models. More of the drug is required to penetrate the core of the complex structure. Now, this is an example of the CyQUANT XTT as an endpoint assay, taking a read after 4 hours of incubation with CyQUANT XTT. So next, we will talk about using the CyQUANT XTT assay in kinetic mode. So as a kinetic assay, the reduction rate of the CyQUANT XTT reagent was monitored over time. Here we have untreated monolayer cells or drug-treated monolayer cells. With the untreated monolayer cells, which is the blue line, the absorbance signal increases over time and eventually plateaus at about 5 hours. This plateau is because all the viable cells have been measured with the CyQUANT XTT reagent. Furthermore, if you look at the drug-treated cells or the dead cells, which is the yellow line, the absorbance signal continues to increase over time, but slowly. This is why it's important to record the background signal and the signal that comes from your media only when treated with the CyQUANT XTT reagent. This background signal that is obtained must be subtracted from your test samples to obtain a true signal from your viable cells. Now, if we look at the reduction rate of the CyQUANT XTT reagent when incubated on untreated spheroids, the blue line, we also see an increase in absorbance signal. However, within 24 hours, we still do not see the signal plateau, indicating that it takes longer for the CyQUANT XTT reagent to be reduced. One thing to keep in mind when studying 3D spheroids is that they are compact as opposed to a monolayer cells, which are very spread out. This is important because with this compact structure, it will take some time for the reagent to enter the center of the compact structure. As similarly to the drug-treated monolayer cells, the drug-treated spheroids presents a very low signal turn-on, confirming the absence of metabolically active cells, which is your yellow line. Now, although the signal from the CyQUANT XTT reagent continues to increase over time with the spheroids, the potency values are very similar. Here we have a table with different incubation times with the CyQUANT XTT reagent and the IC50 values obtained at those incubation times. And you can see within this table the IC50 values are within 30 to high 30 micromolar range, no matter at what time you take a reading with the CyQUANT XTT reagent. And because of this, the CyQUANT XTT cell viability assay can be used for both endpoint and kinetic assay mode. Not only can we quantify the cell viability of spheroids and monolayers of cells, we can also visualize the health of the spheroids by microscopy. These images were obtained on the EVOSM7000 Imaging System on either a flat bottom 96-well plate or on the monolayer cell, 4 monolayer cells, or a NUNC U-bottom 96-well plate for the spheroids. From left to right, we have high concentrations of gambogic acid to low concentrations of gambogic acid. And from top to bottom, we have increasing incubation time with the drug, starting at 2 hours down to 48 hours. So if we look at the top, at the 2 hours of the drug treatment, we can see a tight compact structure of these spheroids, like that of untreated spheroids, which is associated with high cell viability. But as time progresses with the drug treatment, that compact structure is lost, which is seen in the 48-hour images with the high drug treatment at the bottom of this slide. As for the monolayer of cells, the cells ball up and die at high concentrations of drug, which is different to the viable spreading of cells. So also keep in mind the structure of your 2D and 3D cellular models when you're looking at cell viability. So we talked about a photometric cell viability assay. PrestoBlue HS now, though, is a fluorescent cell viability assay. PrestoBlue HS, where HS stands for high sensitivity, is a new and improved viability assay. There are more than 50% reduction in the background and an increase in the signal to background ratio when compared to the original PrestoBlue reagent. Generally, when PrestoBlue is used, it's used for a short incubation time. PrestoBlue HS is an add and read reagent that can be read in both absorbance and fluorescence. However, we highly recommend to use the fluorescence mode because of the higher sensitivity. Now, the main component within PrestoBlue HS is rizazurin, which gets reduced to fluorescent rezarufin when in the presence of viable living cells. Because PrestoBlue HS is a solution-based assay and the rezarufin product gets released into the solution, a top reading is preferred for both monolayer and spheroids of cells. Top reading is best for homogeneous solutions in this fluorescence mode, as mentioned by Adyary earlier in this presentation. Here we have another drug dose response comparison of monolayer cells and spheroids. With this experiment, again, monolayer cells or spheroids were plated at 19 hours to allow the cells to adhere to the bottom of the well and to also allow for that tight, compact structure of spheroids to form. Both models were then treated with gambogic acid, and the cell viability was assessed with PrestoBlue HS at different hours of drug incubation time, which is 2 hours, 19 hours, or 48 hours. As shown within these graphs, as the incubation time increases, again, we see that the gambogic acid, the drug the longer incubation times with gambogic acid, the drug becomes more potent. So additionally, when comparing spheroids and monolayers of cells, gambogic acid is less potent to spheroids than to monolayer cells. The results are very similar to what we saw earlier with the CyQUANT XTT reagent. Here's a summary of the IC50 values obtained from the PrestoBlue HS analysis with the drug-treated monolayer or spheroid models. As I mentioned earlier, high concentrations of gambogic acid are required to kill the 19-hour spheroids when compared to monolayer cells or 2D cultures. Now, specifically with monolayer cells, there is a time-dependent cytotoxic effect from gambogic acid. So the monolayer cells die off faster than your cells within your spheroid structure. We can also look at PrestoBlue HS in kinetic mode. We can monitor the reduction rate of the cells with PrestoBlue HS over time, which is shown in this graph on this slide. There is only one thing to remember when using PrestoBlue HS in kinetic mode. Resirufin can always over-reduce to non-fluorescent dihydroresirufin, which is why at about hour 9 or 10 with this untreated monolayers of cells, the blue line in this graph, the fluorescent signal starts to decrease. If you incubate with PrestoBlue HS too long, the dihydroresirufin will be produced, and you can obtain a false negative. The yellow line is indicative of the absence of metabolically active cells for drug-treated monolayer cells. Similarly, with the CyQUANT XTT reagent, incubating untreated spheroids, the blue line with PrestoBlue HS, there is a slow increase in fluorescent signal over time. The reduction kinetics of PrestoBlue HS on spheroids is slower than that on the monolayer cells, again due to the tight compact structure of cells. And as similarly to the drug-treated monolayer cells, the drug-treated spheroids present a very low signal turn-on, confirming the absence of metabolically active cells. So the PrestoBlue HS assay can be used for both endpoint and kinetic studies. CyQUANT Direct is another fluorescence assay that quantifies cellular proliferation, viability, and cellular cytotoxicity. There are two different components to the CyQUANT Direct assay. There's a nucleic acid dye and a background suppressor. The nucleic acid dye is live cell permeable and concentrates to the nucleus of the cell, and the background suppressor is a cell impermeable reagent that suppresses the green background fluorescence. These two components work well together, that there is no need for a wash step when using them, which makes this assay very simplified. Now, because the fluorescent signal accumulates within the nucleus of the cell, a bottom rate is best for these assays for both 2D and 3D cellular models. Using the CyQUANT Direct assay to evaluate cell viability, similar results are obtained as we saw earlier with the CyQUANT XTT and PrestoBlue HS. A similar experiment was done. As I mentioned before, monolayer cells or spheroids were plated at 19 hours to allow the cells to adhere to the bottom of the well and your spheroid to form. Both models were then treated with gambogic acid, and the cell viability in this case was then assessed with the CyQUANT Direct reagent at different hours of drug incubation time. Again, 2 hours, 19 hours, and 48 hours. So as we saw previously with the other reagents, as incubation time increases with the gambogic acid, the drug becomes more potent. Additionally, when comparing spheroids on the right and monolayers on the left, gambogic acid is less potent to the cells than to the monolayer cells. Using CyQUANT Direct as an endpoint assay, IC50 values were then obtained for the drug-treated monolayer or spheroid models. The cytotoxicity of gambogic acid on monolayers versus spheroids is consistent with the previous results from our XTT and PrestoBlue HS assays. Now, because one of the components of the CyQUANT Direct assay is a background suppressor that continues to enter the cell slowly over time, CyQUANT Direct can only be used as an endpoint assay. As shown here with the untreated monolayer cells, the blue line, after about an hour of incubation with CyQUANT Direct, the fluorescent signal starts to drop and continues to drop dramatically. Now, this drop in signal will provide a false negative. With drug treatment of monolayers of cells, there is no signal of, with drug treatment of monolayer cells, there is little to no signal confirming the absence of DNA after replicating, from replicating cells. A similar decrease in fluorescent signal is seen with untreated spheroids, as shown in this graph with the blue line. So we do prefer that the CyQUANT Direct assay is used only as an endpoint mode for both 2D and 3D cellular models. We recommend this so that you avoid obtaining that false negative response. So far, we have been talking about plate reader, microplate reader assays, and although CyQUANT Direct is a microplate read assay, one advantage of the CyQUANT Direct assay is that it can be imaged as well, given that it's a nucleic acid dye and is most commonly used for high content imaging experiments. So here I show that similar results for cell viability using the CyQUANT Direct can be collecting using either the VarioScan Lux microplate reader or the Cell Insight HCA instruments. On the left is the data collected on the VarioScan Lux microplate reader, and on the right is the data collected on the Cell Insight CX7 HCA platform. Because the HCA takes a half hour or even longer to collect data from a full 96-well plate, and the VarioScan Lux microplate reader only takes a couple of minutes for obtaining data on a full 96-well plate, it would be most beneficial to use the microplate reader for initial screening before moving on to in-depth complex imaging experiments. So here I have a summary of the IC50 values obtained using CyQUANT Direct as a cell viability assay on drug-treated monolayer or spheroid models. As I mentioned earlier, similar results and similar IC50 values are obtained on either the VarioScan Lux microplate reader or on the high-content CX7 HCA platform. So why use a time-consuming complex system like the HCA when you can use a simplified workflow on the VarioScan Lux microplate reader? The only advantage of using the high-content CX7 platform is that you can visualize the structure of the spheroid, and when using the CyQUANT Direct as a cell viability assay, your viable cells become highly fluorescent. Now, the top row of images here are phase contrasts, which allow you to visualize and see the compact structure of the spheroid. The bottom row images are the fluorescent channel for the CyQUANT Direct reagent, labeling viable cells. With these images, you can see how they pair with one another and how the tight core of the spheroid decreases from right to left as the drug concentration increases and the cell viability decreases. Additionally, the untreated and complex structure is labeled with the CyQUANT Direct dye, meaning that those cells contain DNA and are cells that are replicating. Here's a summary table of all the reagents we talked about today in this webinar and their effect on 2D and 3D cellular model systems. No matter what cell viability assay was used, similar IC50 values were obtained at the different drug incubation times, either 2 hours, 19 hours, or 48 hours. And across the board, we saw that as the incubation time increases with gambogic acid, the drug becomes more potent. Additionally, when comparing your spheroids and monolayered cells, gambogic acid is less potent in spheroids than to monolayer cells. Now I will hand off the presentation back to my colleague, Adyary.
Thank you, Leticia. Well, I really hope that we have been able to demonstrate to you guys the suitability of these three assays for these comparative studies between 2D and 3D, or even for just studying or interrogating cellular health functions in 3D cells. Here you can see a summary of the different issues that were discussed by Leticia. We tried to cover all the possible measurements in terms of detection technology. So you had photometric assays in the case of CyQUANT XTT, and then we had two fluorescent assays that can be read with top or bottom reading in the case of PrestoBlue HS with top reading, and in the case of CyQUANT Direct with bottom reading. All of the three assays, as Leticia explained in detail, they can be used for as endpoint measurements. But on the case of kinetic measurements, we recommend to use only CyQUANT XTT and PrestoBlue HS. CyQUANT Direct, we recommend to use only as endpoint measurements. They all are having this kind of atom-read format, so they require really minimal efforts during the workflow, and they have very similar incubation times with the dyes. And in the case of multiplexing compatibility, then obviously the fluorescent assays are superior. And if you are planning to utilize different readouts and combine different endpoints, different cellular functions, interrogate different functions, then obviously utilizing PrestoBlue HS and CyQUANT Direct, even the two of them together would be a better choice. From the perspective of doing orthogonal research and combining these quantitative assays with imaging, then the best choice here is really CyQUANT Direct. You saw the really spectacular images that you can obtain with CyQUANT Direct, and the benefits that then you can combine this quantitative data and correlate with the images that with these really nice phenotypic images that can be obtained. So we hope that you have hopefully learned few essential issues that needs to be taken into account when utilizing these cell health assays with microplate readers. For us, it's really important to convey the idea that we have a number of cell health microplate assays that, even though they were originally developed for 2D cells models, they can be used for 3D spheroids, and they really are a time and cost-efficient approach to assess therapeutic effects of investigational compounds in early drug discovery. We have collected here some of the resources that are available for you on our web pages. We have recently published an application note that covers a number of tips and tricks that can be used for performing high-throughput fluorescent imaging and analysis of spheroids. And that application note, I think we recommend it as a really good tool to start and to know how to approach this transition between 2D and 3D. In that application note, we have a list of 12 reagents or assays that we currently have shown to be effective tools also in 3D using different detection platforms that you can see are listed here. So this hopefully will give you a quite good guideline, and it will really help you navigate this transition between 2D and 3D, and also it will help you performing these comparison studies that we show you today. So we hope that you have been able to follow us through and that you have learned during this hour. Now we have time for taking your questions, and we really thank you for your attention, and we'll be happy to answer any questions. And those that we cannot follow up, then we'll be happy to follow up by email. Thank you.
Thank you, Adyary and Leticia, for your informative presentations. We would love to understand your practices in this field. You will now see a survey pop up on your screen. We appreciate your participation. Okay, we will now move on to the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. All right, let's get started. Our first question is, can you comment on how luminescence measurements for 3D cells performed with plate readers?
Thanks, Michelle. This is a quite interesting question. We didn't have the time to get into luminescence measurements, but generally speaking, luminescence measurements with plate readers are always performed from the top of the plate. So instruments try to collect all the light that is emitted in luminescence reactions. So there are really no basic differences between collecting light in 2D versus 3D systems. So in that sense, the choice has to do more with the assay than with the way in which the light is collected in microplate readers.
Thank you. All right, next question. Can the cell viability reagents presented be used with 3D models grown in Matrigel?
That's a very good question. We get that question very often. So add-and-read solutions or add-and-read solution-based assays such as like PrestoBlue HS or Blue HS and CyQUANT XTT, which we presented here in the webinar, they can be used for 3D models, but the incubation times will have to be optimized. So you generally have to incubate longer than you would with 3D structures not in Matrigel, but keep in mind assays such as CyQUANT LDH, which is where you have to remove the solution from your cells into a brand new plate. In this case, the LDH assay cannot be used for 3D models surrounded by Matrigel, just because of that removal of the media.
Great, thank you. Okay, next question. How can I make sure that the fluorescence or luminescence values that are measured with two different microplate readers are comparable?
That's a great question, and this is a question that we get asked very, very often. So I think it's really important to remember that fluorescent and luminescent measurements are always relative measurements. So they are going to change from one microplate reader to the next one. If you are changing between microplate readers and you want to make sure that you are getting the same performance from one microplate reader to the next one, you have to be able always to measure the ratio between the signal and the background. So that ratio should remain constant if you are comparing different microplate readers that have similar performance. So that is the one thing that you have to keep in mind. You cannot compare directly between one microplate reader readout and another one. Sometimes scaling factors are used so that numbers are larger or numbers are smaller. So just bear in mind that the ratio between the signal and the background on your assay should remain the same if you are performing the same, if you are reading the same plate between different microplate readers with similar performance.
Great, thank you. Okay, next question. How much is the spheroid size a limiting factor for these assays?
That's another great question. So the spheroid size is dependent on the type of plate that you use. You can have we recommend that you start with a low density plating all the way to a high density plating and determine at what is the optimal size for your spheroid that you're actually using and how long of that drug incubation time to use, but not only that, but also how long of your reagent to incubate with. So it's not necessarily the limiting factor is how sensitive your reagent is. So it kind of goes back to your reagent and how sensitive it can detect a number of cells.
Thank you. Next question, how do you measure background signal?
I can answer that. So background signal is always measured on the media or on the buffer that you are actually performing your assay, and it should, of course, have the dye included on that media or buffer. This is really important because, for example, if you are running assays on phenol red containing media, phenol red is known to absorb light at very common excitation wavelengths. So this is something that you have to be aware of, and I didn't have time to get into that, but this is one reason as to why cell-based systems are actually often, monolayers are often measured from the bottom, because then you can exclude the effect of phenol red. But the way that you can establish the background levels is always with the solvent that you're using, media buffer, and then of course having the contribution of the dye in the absence of cells in this case.
Thank you. What's the largest size of spheroids that can be assayed?
So this kind of goes back to the question we had earlier of your reagent itself. And so the more cells that you have, the longer incubation time you're going to have with your reagent. But it also goes back to the size of plate. So we've ran spheroids as high as 10,000 cells per well, but those were with, again, A549 cells or lung cancer cells. If you have a smaller cell type, such as MCF7s, you can actually have more within your well. So you can have 20,000 per well. So the size really is based off of your size of cells. And then the reagent itself, again, you either have to have a longer incubation time for larger spheroids, or you can shrink that incubation time if you go down to, say, 500 cells, so a smaller sample size.
Any other questions, Michelle?
It looks like we have time for one more question. If one is only studying 3D cells and not comparing with 2D models, is top fluorescence enough to quantify fluorescent signals on spheroids?
Yeah, another really good question. In the majority of the assays that we have tested, top fluorescent is pretty sufficient to measure fluorescent signal on spheroids on this model that we discussed during the webinar, but it would need to be optimized and it would need to be compared. It's very, very strongly dependent on the type of assay that you are utilizing and the type of reagent, as Leticia has mentioned several times. So it would need to be optimized, but yes, as a general rule, we would say that top fluorescent might be sufficient for a good portion of the assays out there.
Thank you, Adyary and Leticia, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.